What if I told you the single most powerful thing you could do for your transplants takes 30 seconds, costs almost nothing, and has been practiced by farmers on every continent for generations? Before your seedling ever touches the ground, you coat its roots in a thick, living paste, a slurry teeming with microscopic fungal spores. When you do, something remarkable happens. That plant does not just survive transplanting, it explodes. Root systems double and triple in size. Phosphorus uptake jumps by over 100%. Yields climb by double digits. Under drought conditions, the difference between treated and untreated plants is so dramatic it looks like they belong to different species. This is not some fringe gardening hack. This is not a marketing gimmick from a supplement company. This is a 460 million year old biological partnership that modern agriculture nearly destroyed, and that science is now racing to understand and restore. The technique is called mycorrhizal root dipping, and by the end of this, you will understand exactly why it works, what the numbers actually say, and how to do it yourself in 5 simple steps. Let us start with a question that reframes everything you think you know about how plants grow. What if roots were never meant to work alone? We think of roots as the engine of a plant. They reach into the soil, they absorb water, they pull up nutrients. But here's the thing, roots are actually terrible at their job. They are too thick to fit into the tiny pores where most soil nutrients live. They are too slow to explore the vast volume of soil around them before nutrients get locked up in chemical bonds. And they are too energetically expensive to build in the quantities that would actually be needed to feed a fast-growing plant. Evolution solved this problem a very long time ago, 460 million years ago to be precise. That is when the first land plants formed a partnership with a group of soil fungi that would become the most important symbiosis in the history of terrestrial life. These fungi are called Mycorrhizae, from the Greek words for fungus and root, and they predate the evolution of true roots by roughly 50 million years. Let that sink in. Plants had fungal partners before they had roots. The fungi were the original root system. Every major crop humanity has ever cultivated, wheat, rice, maize, potatoes, tomatoes, beans, peppers, lettuce, onions, every single one of them evolved inside this partnership. Their genomes expect it. Their nutrient uptake pathways are optimized for it. Their stress response systems are calibrated around the assumption that fungal allies are present in the soil. And for most of agricultural history, they were. But then something changed. Over the last century, modern industrial agriculture systematically dismantled the conditions that mycorrhizal fungi need to thrive. Deep tillage shreds the delicate hyphal networks that fungi spend months building through the soil. Synthetic phosphorus fertilizers, applied in massive quantities, send a chemical signal to the plant that suppresses mycorrhizal colonization. After all, why invest carbon in a fungal partner when phosphorus is already flooding in from the surface? Fungicides, applied broadly and often, kill beneficial fungi right alongside the pathogens they target. Bare fallow fields, left without living roots for months at a time, starve mycorrhizal communities of the carbon they need to survive. The result is that much of the world's agricultural soil is now functionally stripped of its mycorrhizal community. And when you go to a nursery and buy a beautiful tomato seedling grown in sterile peat-based potting mix under controlled greenhouse conditions, that plant arrives in your garden with zero mycorrhizal colonization. None. It has never met its fungal partner. You put it in the ground, and it is alone. It has to build its entire root system from scratch in soil that may have only a fraction of the fungal community it evolved to depend on. And we wonder why transplants suffer shock, why they wilt, why they sit there for weeks doing apparently nothing before they finally start growing. They are struggling because they are missing half of their operating system. This is where root dipping comes in, and the beauty of this technique is its simplicity. Before you plant, take the seedling, remove it from its pot, or unwrap its bare roots and dip the entire root ball into a thick slurry. The slurry is a paste made from mycorrhizal fungi spores mixed with water and sometimes a handful of other beneficial ingredients. Coat every root surface and make sure the fungal spores are in direct physical contact with the root hairs. Then plant immediately while the coating is still wet. That is it. The whole process takes less than a minute per plant. You have just skipped months of waiting. Instead of hoping the plant will eventually encounter mycorrhizal spores somewhere in the soil, you have guaranteed contact from the very first moment. The fungal spores germinate within days. Hyphae begin penetrating the root cortex within a week, and within 2 to 4 weeks, the mycorrhizal network is established and actively delivering nutrients. You have given your plant a 460 million-year-old support system on day 1. Root dipping in some form is not new. Nurseries have dipped bare rootstock in plain clay slurry for decades, sometimes centuries. The original purpose was simple and mechanical. Coating exposed roots in a layer of wet clay prevents the delicate root hairs from drying out during transport and handling. Root hairs are microscopically thin, just a single cell wide, and they desiccate within minutes of exposure to air. Once they are dead, the plant has to regrow them entirely, which costs time and energy and delays establishment. A clay slurry coating buys critical hours of protection. This practice was standard in orchards, in forestry, in any operation that involved moving bare root plants from one location to another. The upgrade, the version that changes everything, is adding mycorrhizal fungi inoculant to that slurry. Now you are not just protecting the roots from drying, you are introducing the fungal partner directly onto the root surface in the highest possible concentration at the most critical possible moment, the moment of planting. This is not just a Western innovation. Farmers in Madagascar have been dipping rice seedlings in phosphorus-enriched slurries before transplanting into paddies for generations. Farmers in India practice a similar technique with rice and vegetable seedlings, mixing locally available amendments into a root coating paste. These are established techniques in subsistence agriculture, developed independently by communities who could not afford synthetic inputs and who needed every possible advantage from the biology of their soil. Modern science did not invent this. Modern science only caught up to it. So let us talk about what that science actually says, because the mechanisms behind mycorrhizal root dipping are now understood in extraordinary detail, and they are even more impressive than the traditional practitioners could have guessed. The most fundamental thing mycorrhizal fungi do is extend the reach of the root system. A single plant root is, in soil terms, a blunt instrument. Roots are typically 200 to 2,000 micrometers in diameter, and they can only access nutrients that are within a millimeter or two of their surface. But mycorrhizal hyphae, the thread-like filaments that the fungi extend out from the root, are just 2 to 10 micrometers in diameter. They are 100 times thinner than the finest root hair. This means they can penetrate into soil pores and crevices that are physically impossible for roots to enter. And they do this at an extraordinary scale. A single colonized root system can support a high-fall network that extends the effective absorptive surface area of that root by a factor of 100 or more. Some researchers have measured high-fall densities of 10 to 100 meters of fungal filament per cubic centimeter of soil. The plant is no longer limited to the nutrients within a few millimeters of its roots. It now has access to nutrients across a volume of soil that is orders of magnitude larger. This matters most for phosphorus. Phosphorus is the single most growth-limiting nutrient in the majority of the world's soils, not nitrogen, which gets all the attention. Phosphorus. The reason is that phosphorus, unlike nitrogen, is almost completely immobile in soil. It binds tightly to soil particles and barely moves, even in solution. So a plant root can only access the phosphorus that happens to be right next to it, and once that tiny zone is depleted, the root has to physically grow into new territory to find more. This is energetically expensive and painfully slow. Mycorrhizal hyphae solve this problem elegantly. They extend far beyond the phosphorus depletion zone around each root, tapping into fresh reserves that the root could never reach on its own. And the numbers here are staggering. Research has shown that 75 to 90% of a plant's total phosphorus uptake can be delivered through the mycorrhizal pathway when the symbiosis is well established. The fungal highway is not a supplement to root uptake. It is the primary delivery system. The root's own phosphorus absorption is the backup. We have had it backwards this entire time, but phosphorus is only the beginning. Mycorrhizal fungi also significantly enhance nitrogen uptake, contributing anywhere from 5 to 80% of a plant's nitrogen depending on the system and conditions. They improve uptake of zinc, copper, and other micronutrients that are similarly immobile in soil. And they do something that is critically important in an era of increasing drought. They transport water. Mycorrhizal hyphae can access water held in soil micropores and deeper soil layers that roots cannot reach, and they actively transport that water back to the plant through the hyphal network. This hydraulic function means that mycorrhizal plants maintain higher tissue water content, keep their stomata open longer during dry periods, and continue photosynthesizing when non-mycorrhizal plants have already shut down. In drought conditions, this is the difference between a crop that yields and a crop that fails. The benefits extend even further. Mycorrhizal fungi secrete a remarkable glycoprotein called glomalin, a sticky, persistent molecule that glues soil particles together into stable aggregates. This improves soil structure around the root zone in ways that benefit the plant enormously. Aggregated soil has better porosity, better water infiltration, better aeration, and better resistance to erosion and compaction. The fungi are literally engineering the soil environment to be more hospitable to the plant. And on top of all this, mycorrhizae produce antibacterial and antifungal compounds that offer a degree of biological protection against soil-borne pathogens. Research has documented suppressive effects against Fusarium, Pythium, Rhizoctonia, and various plant parasitic nematodes. The mechanism is not a single magic bullet, but rather a combination of competitive exclusion, an enhanced plant immune response, and direct chemical antagonism. A mycorrhizal plant is not just better fed and better watered, it is better defended. Now I know what some of you are thinking. This all sounds impressive in theory, but what do the actual numbers look like when you test it? Fair question. And the answer, drawn from hundreds of peer-reviewed studies and multiple large-scale meta-analyses, is that the effect is real, consistent, and large. A comprehensive meta-analysis pooling data from hundreds of individual studies found that arbuscular mycorrhizal fungal inoculation produced, on average, a 47% increase in total plant biomass. 47%. That is not a marginal improvement. That is a fundamentally different plant. Another major meta-analysis, looking specifically at shoot and root responses, found a 36% increase in above-ground shoot biomass and a 29% increase in below-ground root biomass. The entire plant grows more, both above and below ground. When researchers looked specifically at field trial results, the kind of data that comes from real farms rather than laboratory pots, a meta-analysis spanning 290 studies found a 23% average yield increase. That is yield, the actual harvestable product, not just plant size. In economic terms, a 23% yield increase from a biological amendment that costs dollars per acre is an extraordinary return on investment. The phosphorus numbers are particularly dramatic. Across studies, arbuscular mycorrhizal fungal inoculation produced a 105% increase in whole plant phosphorus uptake. The plants were absorbing more than twice as much phosphorus from the same soil. This finding alone has massive implications for global agriculture because phosphorus fertilizer is manufactured from rock phosphate, a finite-mined resource that some analysts warn could face supply constraints within decades. If mycorrhizal management can halve the phosphorus fertilizer requirement while maintaining yields, the environmental and economic implications are enormous, and that is exactly what the rice root dipping research shows. Phosphorus-enriched root dipping of rice seedlings, the technique practiced by farmers in Madagascar and India, produced a 10 to 50% yield increase using the same amount of fertilizer, or achieved the same baseline yield while reducing phosphorus fertilizer application by 40 to 60%. Either way, the efficiency gain is transformative. Crop-specific results are equally compelling. Mycorrhizal inoculated strawberry plants produced 17% more fruit compared to uninoculated controls. That is 17% more harvestable strawberries from the same number of plants in the same soil. And under stress conditions, the advantages become even more pronounced. One study examining drought stress responses found that arbuscular mycorrhizal fungal inoculated plants showed up to a 220% increase in biomass at high carbon to nitrogen ratios. 220%. Under the exact conditions where plants struggle most, the mycorrhizal partnership delivers its greatest benefit. This makes evolutionary sense. The symbiosis did not evolve in perfect laboratory conditions. It evolved in the real world, where drought, nutrient limitation, and competition are the norm. The system is designed to shine under pressure. So how do you actually do this? Let me walk you through the process in 5 straightforward steps. Step 1. Get your mycorrhizal inoculant. You want a product that contains arbuscular mycorrhizal fungi, often abbreviated AMF, or sometimes labeled endomycorrhizal fungi. The most commonly available and well-researched species is Rhizophagus irregularis, which you may also see listed under its older name, Glomus intraradices. Many good products contain a blend of multiple endomycorrhizal species, which is fine and may actually be preferable since different species have slightly different strengths. The inoculant typically comes in granular or powder form. Read the label. You want a product that lists a guaranteed spore count, ideally at least a few hundred spores per gram. Avoid products that are vague about their contents or that list only ectomycorrhizal species, which associate with trees and not with vegetables, grains, or most garden plants. Step 2. Make the slurry. Take your mycorrhizal inoculant and mix it with water in a bucket until you get a thick paste-like consistency, something like a thin pancake batter or a clay slip. You want it thick enough to coat the roots and stay on, not so thin that it runs off immediately. The exact ratio depends on your product, but a good starting point is about 2 to 4 tablespoons of granular inoculant per liter of water. Some experienced growers add beneficial extras to the slurry. A splash of liquid kelp extract provides natural growth hormones and micronutrients. A handful of finished compost adds beneficial bacteria that complement the fungi. A small amount of humic acid can help spore germination. These additions are nice but optional. The mycorrhizal inoculant and water alone will work. Step 3. Dip the roots. If you are working with bare root plants, this is simple. Hold the plant by the stem and submerge the entire root system in the slurry, swirling gently to ensure complete coverage. If you are working with seedlings from cell packs or small pots, gently remove the plant and loosen the root ball slightly so the slurry can penetrate between the roots. Then dip and coat. For larger container plants, you may find it easier to pour the slurry over the root ball rather than dunking it. The goal is the same either way: maximum physical contact between the fungal spores and the root surfaces. Every point of contact is a potential colonization site. More contact means faster establishment. Step 4. Plant immediately. Do not let the slurry dry out. The fungal spores need moisture to germinate, and the root hairs need moisture to survive. As soon as the roots are coated, get that plant into its planting hole, backfill with soil, and water in. Speed matters here. This is not a step where you coat everything and then take a lunch break. Coat and plant, one at a time if you have to. The payoff for this urgency is enormous. Step 5, and this one is critically important. Do not add synthetic phosphorus fertilizer at planting time. This seems counterintuitive because we just spent considerable time talking about how mycorrhizae supercharge phosphorus uptake. But here is the biology. When soluble phosphorus is abundant in the soil solution, the plant's root cells detect this and downregulate the signaling pathways that invite mycorrhizal colonization. The plant essentially says, I have enough phosphorus already. I do not need to invest carbon in a fungal partner. High phosphorus suppresses mycorrhizal colonization. This is one of the great ironies of modern agriculture. By dumping synthetic phosphorus fertilizer on our crops, we were actively preventing them from accessing the far more efficient and sustainable mycorrhizal phosphorus delivery system. When you root dip, you are trusting the fungi to deliver the phosphorus. Let them. You can always side dress with other nutrients later if needed, but keep soluble phosphorus away from the root zone during the critical establishment period. Now, understanding why modern gardeners largely forgot about this technique is important because it helps explain the state of our soils and suggests the direction we need to move. The synthetic fertilizer revolution of the mid-20th century fundamentally changed the relationship between farmers and soil biology. When you can pour on granular NPK and get an immediate visible growth response, the slow, invisible work of soil fungi seems irrelevant. Why wait for a biological process when a chemical one gives you results this week? And so the entire agricultural system, from research institutions to extension services to commercial product lines, pivoted toward synthetic inputs. Mycorrhizal biology became an academic curiosity, studied in university labs but largely ignored in practical agriculture. Meanwhile, every major practice of industrial farming was inadvertently destroying mycorrhizal communities. Tillage physically severs hyphal networks. Fungicides kill fungi. Bare fallow eliminates the living roots that mycorrhizae depend on. And excessive phosphorus fertilization suppresses colonization even when spores are present. It was a perfect storm of destruction, and the result is soils across the world that are functionally devoid of their mycorrhizal component. The consequences compound. Plants grown in mycorrhizal depleted soil develop weak, shallow root systems. They are more susceptible to drought because they lack the high-fall water transport network. They are more vulnerable to pathogens because they lack the biological protection that mycorrhizae provide. And they require ever-increasing amounts of synthetic fertilizer because they have lost the biological nutrient delivery system that was supposed to be doing most of the work. It is a dependency cycle. The more synthetic inputs you add, the more you suppress the biology that would have reduced your need for those inputs in the first place. The plants become junkies, and the farmer becomes the dealer. Locked into an expensive and environmentally damaging cycle that degrades soil structure and biological function with every passing season. Root dipping is the fastest way to break this cycle. When you coat roots in mycorrhizal slurry at the moment of planting, you are not waiting for natural recolonization, which can take years in severely degraded soil. You are not hoping that a few surviving spores will find their way to your plant's roots. You are guaranteeing the connection. You are placing the fungal partner directly onto the root surface in high concentration at the exact moment when the plant is most receptive to colonization, during the stress and recovery of transplanting. It is targeted, efficient, and immediate. From that initial inoculation point, the fungal network expands. Within a single growing season, a well-established mycorrhizal plant can serve as a source of inoculum for neighboring plants, gradually rebuilding the soil's fungal community from the point of introduction outward. I want to be honest with you about the limitations because credibility matters more than hype. A major meta-analysis published in 2025 took a hard look at commercial mycorrhizal inoculant products and found that many of them fall short of the results achieved in controlled laboratory studies. The reasons are multiple. Product quality varies wildly across the industry. Some products contain fewer viable spores than claimed. Some contain the wrong species for the target crop. Some have been stored improperly and have reduced viability. The gap between what a high-quality inoculant can do in a research trial and what a random product from a garden center shelf will do in your backyard is real. This means sourcing matters. Buy from reputable suppliers who provide third-party testing data. Look for products with guaranteed spore counts. Check reviews from other growers. The biology works. The question is whether the product you are holding actually contains enough viable biology to deliver. The second important caveat is that results are context-dependent. The most dramatic benefits from mycorrhizal inoculation occur in soils that are low in phosphorus, degraded, disturbed, or otherwise lacking in existing mycorrhizal communities. If you are gardening in rich, undisturbed soil with a long history of organic management and continuous living roots, your soil may already have a thriving mycorrhizal community. Adding more inoculant to soil that is already well colonized may produce minimal additional benefit because the niche is already filled. This does not mean root dipping is useless in good soil. Colonization timing and density still matter at transplanting, but the relative gain will be smaller. Where root dipping produces its most spectacular results is exactly where you would expect, in soils that have been stripped of their fungal community by tilling, chemicals, or long fallow periods. Third, not all plants form mycorrhizal associations. The Brassicaceae family, which includes cabbage, broccoli, kale, cauliflower, Brussels sprouts, radishes, turnips, and mustard greens, is non-mycorrhizal. They evolved alternative nutrient acquisition strategies and do not form partnerships with arbuscular mycorrhizal fungi. Beets and spinach from the Amaranthaceae family are similarly non-mycorrhizal. Dipping the roots of these plants in mycorrhizal slurry will not harm them, but it will not help them either. Save your inoculant for the plants that can actually use it, which is the vast majority of everything else in your garden. And fourth, be realistic about the magnitude of results in a home garden context. The 47% biomass increase and 23% yield increase figures come from averaging across hundreds of studies, many of which were conducted in controlled conditions with optimized inoculants and precisely managed variables. In a real garden, with variable soil, weather, and management, your results will vary. You should expect meaningful, visible improvements in plant vigor, root development, drought tolerance, and yield. You should not expect miracles. A healthy plant in good soil, with adequate water and reasonable fertility, is going to grow well regardless. Mycorrhizal inoculation helps plants grow better, establishes them faster, and makes them more resilient when conditions turn challenging. That is a substantial and worthwhile benefit. It is not magic. With all of that said, let us zoom out and appreciate the bigger picture. 460 million years of coevolution built this system. Long before there were farmers, long before there were gardens, long before there were roots as we know them, fungi and plants were already partners. The mycorrhizal symbiosis is not some optional add-on. It is the foundational biological infrastructure of terrestrial plant life. Every forest on Earth runs on this network. Every grassland, every meadow, every wild ecosystem depends on the underground fungal internet connecting plant to plant, shuttling nutrients from where they are abundant to where they are needed, knitting the soil into a living, breathing, functioning whole. When we disrupted this system with industrial agriculture, we did not just lose a nice to have. We lost the operating system that plants evolved to depend on. And we have been trying to replace it ever since with expensive, energy-intensive, environmentally damaging synthetic substitutes that produce diminishing returns while degrading the very soil they are supposed to be feeding. Root dipping is not the whole solution. Rebuilding soil mycorrhizal communities at scale requires changes to tillage practices, fertilizer management, cover cropping, and crop rotation. It requires a fundamental shift in how we think about soil, from treating it as an inert medium that holds plants up while we pour chemicals on them, to recognizing it as a living ecosystem that, if properly supported, does most of the heavy lifting for us. But root dipping is the fastest, simplest, most accessible entry point into that shift. It is something any gardener can do today, with minimal cost and minimal effort, and see real results within a single growing season. Here is what I want you to do. This spring, buy a $10 bag of mycorrhizal inoculant from a reputable source. When you transplant your tomatoes, your peppers, your squash, your cucumbers, whatever you are growing, split them into 2 groups. Dip one group in mycorrhizal slurry before planting. Plant the other group normally, without treatment. Same soil, same water, same sunlight, same care. Then watch. Watch how quickly the dipped transplants establish. Watch how much more vigorously they grow through the first month. Watch how they handle the first dry spell of summer. And when harvest comes, weigh the fruit. Count the peppers. Measure the yield. You will see the difference. It will not be subtle. 30 seconds of root dipping, a few dollars of inoculant, and you have just reconnected your plants to a symbiotic network that has been sustaining life on land since before the dinosaurs existed. 460 million years of evolution built this system. All you have to do is reintroduce it. Your plants already know what to do with it. They have been waiting.